We all began life as a single cell. So the question is, how do we have structures like a brain, heart, stomach, and skeleton? Well, it's time to take a trip back to the fetus days. After fertilization and rapid cell division called cleavage, a blastocyst is formed. This hollow developing structure has something special called the inner cell mass. The inner cell mass forms body structures and systems, and is what allows the single cell to become neurons, blood cells, muscle cells, and more through the process of differentiation, where each gets a different structure and function. This normally happens in embryos only, but we have special cells called stem cells, which are undifferentiated and can become any cell type. They self-renew through mitosis and can be classified as either embryonic, which are pluripotent, having any differentiation capability, or somatic, which are unipotent or multipotent, having location-specific differentiation and are found in body tissue. We all have somatic stem cells, but they are not naturally pluripotent, have limited division in vitro, and normally differentiate into specific cell types. There is, however, a lower chance of immune rejection when using a patient's own cells. To reduce somatic stem cell drawback, scientists have experimented with induced pluripotent stem cells, or iPSCs, which are reprogrammed to become pluripotent and generate the three germ layers. Together, the endoderm, mesoderm, and ectoderm create an entire human being. However, these techniques are still in experimental stages. So how can we reprogram stem cells? We know that differentiation is controlled by special proteins called transcription factors, which when added to somatic cells could make them pluripotent again. These proteins control which genes are expressed and help cells maintain an undifferentiated state like a light switch, turning genes on and off. They work by attaching to a site on the DNA so another enzyme, RNA polymerase, can bind. DNA is transcribed into messenger RNA, which then travels to the cytoplasm for protein synthesis. Proteins formed serve many functions in cells. In 2006, 4 transcription factors were found to work, but 1 may promote cancerous tumors. So a 2007 study found 4 more to be successful. I guess the big question here is, what's the point? The magic about induced pluripotent stem cells is that instead of using drugs with side effects, a patient's own body could be used to heal them. With this technology, we can perform organ transplants, treat diseases like diabetes, Alzheimer's, Parkinson's, ALS, and more, and also conduct drug testing with cells generated from a patient's own body, which lowers chance of rejection by the immune system. We can regenerate tissues, create specific cell types, and produce any cell type, all with an iPSC. And it is genetic engineering that has made such technology possible. So the scientists of tomorrow can continue their magic for happier health and a happier world.